Hello, welcome to another video of the add-on course in instrumentation in life sciences. Today in this video, we are going to see about the electroblotting technique. Okay, so welcome myself, Mr. Ashutosh Upadhyay, will be teaching, talking about the Western blotting technique. Okay, so let's start. Okay, so first of all, let's see what is blotting. Okay, so blotting is the transferring of DNA, RNA, and protein onto a carrier. This carrier may be, there may be different carriers that may be used to blot the DNA or RNA. Okay, like nitrocellulose membrane or nylon membrane. Okay, so these are the different kinds of carriers that can be used to blot the DNA, RNA or protein bands onto a membrane made up of this material. Okay, there is certain chemically reactive membrane also that is used in case of RNA that we are going to see in the further techniques. Okay, so let's see one by one. The blotting techniques majorly is divided into three different types based on the biomolecule that you are separating. The first biomolecule that is separated is DNA, and therefore the blotting is called as southern blotting. The next one is the blotting of RNA, and it is called as northern blotting. And next one is the blotting of proteins, and it is called as western blotting. Okay, so let's see first about southern blotting. Southern blotting is one of the very important techniques that was discovered by scientist Edwin M. Southern in 1975, and this is one of the technique that has no alternative till date. Okay, so here what we do in southern blotting, we detect the DNA molecule. Okay, DNA molecule or a gene from different species. Okay, we are going to detect a DNA molecule or gene from different species using the southern blotting technique. Okay, so the DNA can be detected by a single gene or a part of larger piece of DNA such as viral genome that can be easily detected by using southern blotting. Okay, so what is done in southern blotting? There are two different techniques. First is agarose gel electrophoresis, which is used for separation of the DNA molecule, and then this DNA molecule that is separated on the agarose gel is then blotted onto a filter membrane, a specific filter membrane that is used for probe hybridization. Now this membrane, as I told you, can be made of nylon, that is artificially synthesized, or it can be made up of nitrocellulose. Okay, so these two membranes are generally used in case of southern blotting. Okay, so therefore this method is called hybridization because we are hybridizing a probe molecule in order to detect a DNA molecule here. Okay, so generally the probe molecule that is used is a single stranded target DNA. This single stranded DNA binds to a sequence in the genome of an organism in which we want to detect a particular DNA or a gene. Okay, so how this happens, let's see. Okay, so what happens, the mixture of molecule is first separated. Okay, so the mixture of molecule is separated by using agarose gel electrophoresis, as explained in my first video of this add-on course, that is the instrumentation for molecular biology. Okay, now the molecules are immobilized on the matrix. Okay, suppose this is the agarose gel, and onto this agarose gel what you have done, you have separated the DNA molecule. Okay, now this separated DNA molecule should be transferred onto a particular membrane. Okay, so we are going to take a membrane, that is nylon membrane or nitrocellulose membrane. This membrane is generally positively charged. Okay, since you know DNA is negatively charged, it easily blots onto this membrane. Now this membrane is blotted onto this using a electroblotting assembly. Okay, this electroblotting assembly is a vertical electrophoresis unit in which you prepare a sandwich of agarose gel and membrane. Okay, one side there is agarose gel and in other side there is membrane. Okay, and electricity is produced. Okay, now suppose this side is the agarose gel. Okay, suppose in this electrophoresis unit the side to the left is agarose and the side to the right is the actually membrane, that is nylon membrane or nitrocellulose membrane. Okay, so what is going to happen, this will be positive charge and this will be negative charge, and this electroblotting is done for around 2 hours. Okay, so what will happen, whatever the DNA bands that are negatively charged which are separated on the agarose gel are forced to blot onto the membrane. Okay, so this is how the blotting of the DNA happens onto the membrane in case of southern blotting. Okay, so the probe added to the matrix binds to the molecule, and any unbounded probes are then removed by subsequent washing of different kinds of buffers. Okay, now this probes is connected to the target molecule. What happens now, after the electroblotting process in southern blotting, the membranes have blotted onto the nitrocellulose membrane or nylon membrane. Okay, then you have to treat this with UV, okay, ultraviolet rays. Okay, ultraviolet ray treatment, what it does, it immobilizes the DNA strand onto the membrane, and after that baking is done at 70 degrees Celsius, which fixes the DNA bands onto the nitrocellulose membrane, so that when we are going to do the detection, then it is easy for us to see the bands or check out the bands which is present in that. Okay, and we are going to treat this with the probe solution, and specific probe is going to bind to the specific site. Suppose at this site our probe is binding, and this probe is attached with a certain sequence or certain detection procedure, for example isotopic detection and non-isotopic detection. Due to this, what is going to happen, due to this it is going to show us the fluorescence. Okay, if it is non-isotopic detection, it is going to show us the fluorescence, and if it is radio detection, then we are using a radioactive probe, and it is going to show us the radioactive fluorescence. Okay, so this is how this happens in the southern blotting case. You can see here, this is how the electrophoresis is done, and then electroblotting apparatus is shown to you. It is a vertical apparatus where you place the agarose gel and membrane side by side. Okay, and membrane is placed on the positive side and agarose gel is placed on the negative side, so the DNA will be blotted into the membrane. Okay, so this is the figure that it shows. Okay, so you can see the sandwich showing here properly. Here there is electro transfer buffer, and then gel is placed onto which the membrane is placed. Okay, and through this is a traditional assembly which is shown to you here, where traditionally by capillary electrophoresis the membrane is going to absorb the DNA bands from the agarose gel. Okay, but nowadays whatever the electroblotting machinery that is coming up is having the electrophoretic mobility, so that the DNA bands are blotted onto the membrane fast. Okay, it requires around 2 hours, but in case of this traditional electroblotting, this is a traditional electroblotting assembly, it takes place around 24 hours. Okay, it requires 24 hours to blot the DNA, whereas in the latest electroblotting machines it requires less time, because the blotting is done instantly. Okay, within 1.25 to 2 hours. Okay, so these are the steps in southern blotting. Whatever the DNA that is that you are going to check for the presence of a particular gene, you have to first digest that gene. Okay, and digestion of DNA is done by restriction enzymes. Okay, due to this, what happens, the whole DNA from a organism is digested into different pieces. Suppose here 3 pieces are done. Okay, now here what happens, suppose our fragment of interest is present in any one of the gene. Okay, so what I am going to do first, I will be electrophoresing these fragments. Okay, so after electrophoresing, what will happen, these fragments are moved on to the agarose gel. Okay, the fragments are separated depending on the size on the agarose gel. Okay, and I am going to use this electro transfer assembly, and in this electro transfer assembly, see this, due to the capillary action, our gel is placed here. This is our gel, and the DNA bands, these DNA bands are blotted onto the membrane that is placed above the gel. Okay, so this is how the membrane looks like. Okay, so our 3 DNA fragments have been blotted onto the membrane. Okay, now what is done, now you are going to add the detection system. Okay, there are 2 types of detection system. First detection system is isotopic detection. In isotopic detection, what is done, you use a radioactive probe. Okay, so due to this, what happens, this radioactive probe easily binds to the fragment of interest, and then by radio tracer technique or autoradiography, you are able to detect that particular band easily. Okay, but in case of non radioactive detection, what is done, you add a particular molecule to the probe. Suppose this is a probe sequence, and you add a molecule such as biotin to it. Okay, so biotin is what, biotin is vitamin H. Okay, and this biotin labeled probe is added to the membrane which is already baked. Okay, due to this, what will happen, this biotin probe is going to bind at this position, and this biotin is added with another set of enzyme called as HRP streptavidin complex. HRP streptavidin complex. Okay, now this streptavidin is having an affinity for biotin. Okay, so naturally streptavidin is going to bind to the biotin, and it is attached with an enzyme called as HRP. So when you add a substrate such as TMBH202, this HRP enzyme breaks the peroxide here and forms TMBO. Okay, now this TMBO is blue in color, and therefore a blue color is formed, and we can detect the band onto the southern blotting. Okay, so this is how the result is going to look on the X-ray film, and if you are doing it non-isotopic detection, then onto the membrane you are going to see a band here. Now let's see how it looks like when blotted. Okay, so first step is here, you can see separation of the DNA bands onto the agarose gel. After separation, it will be electroblotted. Okay, so here it shows electroblotting. Okay, so this is how electroblotting happens. After electroblotting, the upper membrane that is shown over here is having the DNA bands. Okay, so these bands are added with the radio labeled probe and incubated along with the membrane. In case of non-radio labeled detection, you use different kinds of detection. For example, biotin-related probe detection, then deoxygenin detection. Okay, so wherever the DNA band is present, that DNA band is easily visualized here in this case. Okay, so this is how the southern blotting happens, or southern blotting process takes place. Okay, now let's see how the summary. First of all, you have to extract the DNA, then restricted DNA sample is done, then it is sorted by electrophoresis, that is agarose gel electrophoresis. Okay, after electrophoresis, the denatured DNA is transferred into the nitrocellulose membrane or nylon membrane, and excess DNA is blocked using blocking buffer. Okay, and after the blocking, then washing off is done along with the help of probe, and after that autoradiography or radio tracers technique is used for the further detection. Okay, so there are different ways through which the southern blotting technique is used. It is used in gene discovery, then mapping, evolution, developmental studies, diagnosis, then forensic sciences. It is used as a DNA fingerprinting for detection of different culprits. Okay, identification of transferred gene is also done. So therefore southern blotting is very important in a process that is cloning. Okay, so genetic engineering has a very big importance of southern blotting. Okay, so now let's see the next type of blotting, that is RNA blotting or northern blotting. Okay, so RNA is reactive than DNA, and RNA is always present in double stranded or single stranded form. Okay, so it is blotted onto a membrane. Okay, now this membrane is not a regular membrane, it is not a nitrocellulose membrane or nylon membrane. It is blotted onto a specially designed chemical paper that is called as diazotized paper. Okay, so this is benzoxymethyl diazotized paper, that is Wattmann filter paper 540. Okay, this filter paper is used as a membrane that is first of all made reactive, and then the RNA is blotted onto this kind of membrane. Okay, so this technique was first time discovered by James Elwine and George Stark at Stanford University 1979. Okay, just to do the homology, this technique was given the name northern blotting. Okay, so let's see how it happens. You can see RNA isolated from several biological sample is actually like this. Okay, it is single stranded form. Okay, so it is more reactive, and it is more difficult to transfer. Okay, now here also simply RNA is labeled, and then the RNA electrophoresis is done onto the agarose gel, and after the electrophoresis, the gel is taken and then blotted onto the different kind of paper. Okay, I already told you the membrane which is used is a diazotized paper, that is diisobenzoxymethyl paper. Okay, so this is a special paper that is used for RNA blotting only. Okay, because RNA is reactive it gets blotted onto this kind of paper, which is made from Wattmann filter 540. Okay, generally this paper is used for northern blotting, but sometimes nylon membrane or nitrocellulose filter paper is also used. Okay, but this is more preferred because RNA binds properly onto this membrane. Okay, so here you can see how the electroblotting process takes place. Uh, at the starting, the sandwich is prepared, below the gel is kept, and onto that the membrane is kept, or this filter paper, reactive filter paper is kept, and then some weight is kept on that, and due to the capillary action, the RNA bands are get absorbed onto the membrane like this. Okay, so the RNA bands is transferred to the filter paper. Okay, this chemically reactive filter paper or nitrocellulose membrane. Okay, so this is how the electroblotting is happening in case of RNA. Okay, after that you can go for a radioactive detection and uh, or a non radioactive detection. Okay, you can see the radioactive detection is done. You can see RNA blot corresponds to the sequence of interest. Here the RNA solution is poured with the hybridization buffer, which has labeled probes. Okay, so this solution is mixed. These are the probes. Okay, these are radio labeled probes. Okay, now what happens, these probes binds at a very specific location or the strand which is having a complementary sequence only, means in this whole number of RNA strands, the RNA which is having a complementary strand to our probe is going to bind there. Okay, and our probe is radio labeled, you know it. Okay, so it binds at a specific sequence only where it finds the complementary sequence. Okay, and this probe is easily detected using autoradiographer machine, and then you can detect the fragment of interest. You can see autoradiogram is shown to you here. This is a marker lane, and these are the samples A, B, and C that is easily detected using the autoradiography machine. Okay, so this is how the electroblotting happens in the RNA case. Now let's see how or what are the applications of their the applications are there for RNA blotting. Okay, it is helpful in gene expression and the levels of mRNA. At a particular time, whatever the mRNA level a cell is having, that is detected. Okay, then mRNA transcript size is also detected. Then mRNA degradation is also detected. RNA splicing, half life, and it is often used to check the RNA silencing using transgenic mice also. Okay, so these are the various applications of northern blotting. Now let's see the third blotting, that is Western blotting. Okay, Western blotting is also called as protein blotting. It was discovered in 1981 by Taubin. Taubin. Okay, Taubin et al. It was discovered by this scientist. Okay, here you detect the protein. Okay, so obviously you are going to separate the protein first. Okay, so Western blotting is used for detection of protein. Okay, now let's see how it happens. First of all, for the separation of protein, the proteins has to be separated or electrophoresed onto a gel. And the gel that is used for separation of protein is SDS page gel or polyacrylamide gel electrophoresis. Okay, so here what is done, the electrophoresis, the mixture of protein samples is loaded onto the wells in the SDS page, and after the electrophoresis, the protein sample separates on the basis of their charge. Okay, now after the separation of the protein, what is done, the electroblotting is done. Okay, so similarly the electroblotting is done. You can use a electroblotting assembly also. You can do the blotting traditionally also. Here the nitrocellulose membrane is used. Okay, or nylon membrane is used. Okay, okay, what is done, the gel is placed adjacent to the probe, and in the electric current, what happens, the proteins are blotted onto the membrane. Okay, like earlier techniques, these proteins are immobilized. Here the detection is not done using probes, the detection is using the antibodies. Okay, the detection is using the antibodies sets. Okay, so now you know there are different kinds of antibodies: primary antibody, secondary antibody. Okay, so the primary antibody first of all binds to the protein band of interest. Suppose this is the band of interest, the primary antibody binds at this position. Okay, then afterwards what happens, uh, you add the secondary antibody. Okay, now the secondary antibody which you are adding is actually binded with the enzyme. Okay, you can see that enzyme is called as reporter enzyme. Okay, here it is written over here. Okay, reporter enzyme. Okay, So you can see the protein, target protein is present onto the immobilized membrane, that is nitrocellulose or nylon membrane. Our antibody, first antibody binds at that position. Okay. Now the first antibody is binded with the secondary antibody here, you can see, and that secondary antibody is attached with this enzyme that is called as reporter enzyme. Okay, so the reporter enzyme can be HRP, horseradish peroxidase, or it can be alkaline ALK, alkaline phosphatase. Okay, so what is this enzyme does? You add a substrate for this enzyme. Okay, so generally we use HRP and we add TMB H2O2 O2 as substrate. Okay, so HRP, that is a peroxidase enzyme, breaks this H2O2 into TMBO, which is a red colored or which is a blue colored product. Okay, and this blue colored product is easily visible to us onto the membrane. Okay, so you can see this is how the detection is done. The Western blotting is having lots of applications. It is the biggest and important confirmatory test for HIV virus or AIDS. Then it is used for bovine spongiform encephalopathy disease. Then some forms of Lyme disease is also detected using the Western blotting. Okay, so this is how you do a detection using the electroblotting assembly. So this was all about the video today. Thank you for watching.